here doing this presentation together with the postdoc that we hired on this project, Michelangelo La Spina, and he will also take over from me for the second half of this presentation. So basically this project, go back. This project was set up because aphids are one of the most important problems in greenhouse ornamentals. After thrips, basically, aphids are one of the biggest problems, and especially foxglove aphids, because it seems that there's a lack of effective biocontrol agents. The foxglove aphids are particular problem because there seem the biocontrol agents that are recommended for this aphid seem to lack efficacy. Now currently neonicotinoid insecticides provide a certain level of control and are easily integrated into an IPM program. However, we all know that its use is being restricted by retailers now, and alternative measures are needed. So on the next slide, I will show you the objectives of the project. So the main goal is to provide effective pest management strategies for problem aphids, and in this case it's the foxglove aphids, in greenhouse ornamentals, and especially develop methods that are compatible with existing biological control systems. This is important, especially because we have to use biocontrol for thrips. So anything that we do in addition to that will have to be compatible with thrips biocontrol. We broke the project down into 3 years. The original intention was in year 1 to develop a better understanding of the behavior and life cycle of these foxglove aphids and their interactions with natural enemies in greenhouse settings. Then in year 2, we wanted to investigate potential new biocontrol agents. Could be pathogens, predators, or parasitoids, to identify effective candidates to use in a better aphid management program. And then in year 3, the intention is to determine the compatibility of different candidates, biocontrol organisms, with existing controls, including natural enemies and insecticides, and develop recommendations for their use. Also assess the efficacy of these approaches on different scales, and engage with the industry, especially in Ontario and Quebec, but basically Canada-wide, to transfer these new technologies. So if we go to the next slide, we had a few challenges in our first year. Basically, when we got the go-ahead, we advertised for a postdoc, and it took quite a long time to find a suitable postdoc. So in the meantime, we hired a research assistant to start some of the work, and Sarah Jendricic, our OMAFRA specialist, also helped out with the project. And we advanced some work on potential biocontrol agents that was originally planned for 2016-17. Now we have hired a wonderful postdoc, Michelangelo, and he started in January, and he is on track to go ahead with the project. So basically, I'm glad to say that the project is on track. Next slide. So the revised project objectives are basically a rearrangement of year 1 and year 2. And on the next slide, I will address what we have done in the past year. So basically, we started to develop a better understanding of aphid interactions with natural enemies. The main questions we addressed was, where do foxglove aphids go in summer? Because they tend to be more of a pest in the cooler period, especially spring and fall, and tend to disappear in summer. Now, of course, they don't disappear. They don't go on vacation or anything. So we needed to know, where do they go? And the second question that we concentrated on was, why does the currently recommended parasitoid, Aphid urvi, provide inadequate control? Then we also investigated different biocontrol agents, of which Dalosia coriaria, previously named Aphid coriaria, was one, entomopathogenic microbes and a predatory mite, and I will present on all these things. So the first question, where do Foxglove aphids go in summer? We observed, and growers will probably be able to tell me this as well, that Foxglove aphids show a preference for cooler places during the summer. And we really notice them infecting the leaves of, for example, pansies, where the leaves are almost buried. Those bottom leaves are almost buried in the soil, and that's where they tend to hang out. On sweet pepper, they also tend to migrate down. So we think this gives them a refuge from natural enemies. And normally these leaves are also not scouted for aphids, so it's easy to miss a budding infestation. So we realize that Foxglove aphid preference within the plant probably also depend on plant species and plant developmental stage. So in the next year we'll also further look into this. On the next slide. The second question was, why does Aphidius urvi, the parasitoid on the picture, provide inadequate control? And we had two hypotheses. The first one is that Aphidius urvi searching behavior might not be adapted to find Foxglove aphids. And the second one was that Foxglove aphids also tend to escape parasitism by dropping off the plant when they're disturbed. In the next slide. Our first hypothesis was really based on work done by Sarah Jędrzejczyk on Aphidolitis, where she saw that Aphidolitis, a gall midge, which also attacks aphids, tends to attack aphids on the growing points of the plants and tends to ignore aphids on the bottom of the plant. The next slide. We put a lot of plants in a greenhouse, sat down, released a lot of Aphidius urvi, and each had our eyes on a aphid-infested plant, and basically noted where the Aphidius urvi tended to hang out. So in this graph you can see the middle and the top of the plants had way more Aphidius urvi than the bottom of the plant. So Aphidius urvi tends to prefer the top and middle strata. And this was done on a sweet pepper seedling, which is a plant with a nice stratification. This graph describes Aphidius urvi landing on aphid-infested leaves. So you can see that they liked to land, were more likely to land on aphid-infested leaves when they were on the top of the plant than on the bottom leaf. Now the next slide. However, we did not see the same trend in parasitation. So they seem to like more to parasite aphids on the bottom leaves. The first hypothesis, we probably have to reject it. Although the Aphidius like to hang out more on the top of the leaves, it also seems that they like to walk around more on the top of the leaves and parasite a little bit less. So next slide. The second hypothesis was the aphids dropping behavior. So what we definitely saw in this experiment is that Aphidius urvi triggers defensive dropping. In the 20 minutes that we observed the plants, 65% of the plants had all the aphids on one leaf scatter in response to an urvi attack, and they would either run away or drop off the plant. So in the next experiment, what we're going to do, oops, go back. We're going to compare, so if the aphids drop and scatter, the risk is that you are only spreading your problem if they're not being parasitized, and that you will end up with more plants that are infected with aphids. So the next steps will be to compare the spread of aphids and the efficacy of aphid control using different predators with different attack strategies. So we know Aphidius urvi disrupts the aphids a lot, while Aphidolitis epidemisa, a gall midge, tends to be a very sneaky predator. On this slide, I put that we were also going to look at Aphidius matricariae, which also has that scattering behavior, but doesn't parasitize as much. But we are going to replace this treatment by using a predatory mite, especially because we did not get the Aphidius matricariae from our supplier, unfortunately. So on the next slide. Like I said, we also investigated new biocontrol agents, the Locia coriaria and entomopathogenic microbes in the predatory mite. And I will show you on the next slides what came out of that. So having the aphids drop off the plant really made us think, like, what if we add a predator that's based on the soil surface, like, for example, the Locia coriaria, with the idea that this predator could maybe clean up all the aphids that jumped off the plant. Also, our observation that foxglove aphids migrate to the lower leaves in the soil level in summer would make this a logical choice. However, in our bioassay, we saw absolutely no predation. Even after 3 days of having the aphids there in with the Locia, we saw no predation. And to test that they were actually going to the top of the leaf and the bottom of the leaf, we put some dried cat food, which is something they're reared on the top of the leaf, and immediately they started feeding. So they were active, they were just not recognizing the aphids as prey. So it was a nice idea, but didn't work. Then we tested several biopesticides. Amongst those were Grandivo, which is what's it called? A Chromobacterium, Met52, a fungus based on Metarhizium, and another experimental strain of Lecanicillium lecanii. And in the graph you can see the first one is 1 week after application of the treatments, that Met52 already gave significantly better control of the aphids, and especially after 2 weeks it reduced the aphid populations significantly more than any of the others. Next slide. And it's always nice to show pictures of dead aphids. It's always encouraging to see this. So these are aphids killed by Grandivo, Lecanicillium, and Met52. Next slide. And the last predator that we looked at was a generalist predatory mite. This was actually collected on the Finland campus, and we have been looking at its predation behavior for several pests. In the graph, you can see that this predatory mite, in a little pop bottle assay, like you see on the picture, greatly reduced the number of nymphs and the number of total aphids on the plants. The green bars are without mites, and the red bars are with mites. It will eat adults as well. So again, this is a very interesting option to include in further development of an foxglove aphid IPM program. Next slide. So now I will hand it over to Michelangelo, and he will tell you about his plans for the coming 2 years.
Good afternoon. My idea is to continue the objectives of the project and to conclude the first objective, to try to develop a better understanding. And I will focus also in something that it's evident, but sometimes we don't check on it. Is make a greenhouse scouting, looking for where are the aphids, because it's a project for ornamental crops. Each crop is different. We'll see where they are living, in the top, in the bottom, the top of the plant, where exactly we can found it, and check also the natural enemies that are associated in case. We are going to check also to make surveys around the greenhouses, to for the same reason, looking for the associated natural enemies. So to continue on that, we are going to study some biological control agents, and we are going to investigate those found on this survey, but also those that are used that we found in the literature. So we are going to start right now some experiments with lacewings, overflies, and different predators that can help us to control the Foxglove aphids. And also to check, of course, the compatibility with the biological control agent that we are using right now. So please, the next slide. And for the next year, we are going to follow the schedule, and we are going to determine the compatibility of the candidate that we are going to find with the existing one to control if the interactions are positive or negative, because maybe they can make intra-guild predation. So we are going to study that problem and design the best strategies for to release them in the houses. And that we are going to start to transfer in a small and medium large-scale study in greenhouses that we have here in Mainland, but also in the future we are going to study that, engaged with the industry of Ontario and Quebec, with many wood growers and the biological control companies. Please, next slide. And during this time, we are going to transfer our knowledge and technology developed here in different places for a different kind of public, for the growers, in the Canadian Greenhouse Conference and during the Greenhouse Canada Grower Day. And also we are going to explain our results in the international scientific conferences, and also of course in the articles, workshops, and this is important in demonstration with in trials and station with our colleagues in UMAFRA and ICTO. Till now, we make only one presentation last year in the Greenhouse Conference, so we expect to contribute more in this transfer. So please, next slide, please. And we would like to thank all the people engaged in our project, mainly people from OMAFRA, like Sarah Jandis, our technician Rebecca, and Taro, our director, Michael, and another technician, Ashley, and the companies that collaborate in the project. Many thanks for your attention.
Any questions for Rose and company?
Hey Rose, it's Jamie here. I was wondering, with the biopesticides, the MET52 showed the best efficacy. How was it applied to the aphid or to the plant?
The MET52 was applied as a spray.
As a spray?
Yeah.
At the label rate?
I will have to ask Taro. I'm assuming so.
Okay.
I do have a question finally. It's Caroline.
Okay, good.
Rose, was it applied only once?
The biopesticides?
Yep.
Yes, that was only one application.
Okay, so that's...
Well, it might even be better if you have maybe 2 applications in a row or something.
But that's pretty interesting.
Yeah.
Thank you.
Yeah, no problem.
Rose, do you consider the economics of any of this? Is that an issue at this point, or are you just addressing the biology?
Well, of course, the final program has to be economically feasible. I mean, if we say, like, use all these enemies, natural enemies, and introduce them 10 times, and of course that won't be a viable program. So our goal is really to have something that growers can use, and that's not too expensive. compared to what they have to do now.
Good, understood. So I think, unless there are other questions, I'm holding here. Does anyone have a question then?
I have another one. If
Okay, Jamie.
I'm interested in the defensive dropping you talked about with the when the Irvi was in the presence of the Foxglove aphid, it would drop off the plant or run away.
Correct.
Where would it go? Like, would it drop into the soil then?
We don't know exactly. For the reason we would like to continue our experiments to see where they go. Normally, the idea is to run away to another plant to continue the feeding process, but we don't know. So it's something that we have to continue to study.
Yeah. Well, from one experiment that we just completed, is that we saw a lot of them really drop. So we put a cup under the leaf and then released a parasitoid, and it's just raining aphids. And some of them just try to run away as well to places where there's no parasitoids. So presumably there is a cost to stopping feeding and a risk of predation that they can run into on like the soil surface or trying to run up another plant. But again, there's also the risk that you're, by dropping, you're only spreading the problem.
Right.
And in Dave Gillespie's lab with Matricariae, they actually saw this happening, that the aphids actually spread.
Okay. So so that's not something you'd like to see.
Not necessarily. If they don't die where they drop, then yeah, that's not something you would like to see.
This is Sarah here. Something we might look at in the future is what type of bench covers, if that influences anything. If, the mesh type, they just fall through underneath and die, or if it's a smooth plastic tarp or cover or bench top, then if there's a higher chance of them spreading, that would be interesting.
Yeah, it would be something we could include in our greenhouse scouting, see if anything falls under mesh benches or